The overall goal of this procedure is to demonstrate how to use zinc-finger domains to deliver functional proteins, such as green fluorescent protein, into mammalian cells. This is accomplished by first generating an expression plasmid containing the emerald green fluorescent protein, MGFP, translationally fused to zinc-finger domains. The second step is to express the fluorescent fusion protein in bacteria. Next, the bacterial cells are lysed and the fluorescent fusion protein is purified. The final step is to incubate mammalian cells with the purified fluorescent fusion protein to allow uptake. Ultimately, the fluorescence of treated cells can be quantified by flow cytometry to determine the efficiency of fusion protein delivery into the cells.
The main advantage of ZIF over other protein transduction domain, like HIV-derived tight peptide or polyarginine, is that it can facilitate high level of cytosolic delivery without compromising the activity of fused enzymatic cargo.
To begin, PCR amplify the emerald GFP or MGFP sequence from a plasmid according to the text protocol. Purify the PCR product using a gel extraction kit and then quantify the DNA concentration using a spectrophotometer. Next, obtain a PET28 expression vector containing the sequence of alanine-substituted 2-finger zinc-finger or 2F-ZIF domains. Digest the vector and the